Hello, good afternoon. Welcome to the third International Plant Seminar of the series, of the spring series that's organized by NC State. Please, again, as in previous seminars, tell us through the chat where are you joining from, and please post your questions in the QA link, okay? And we'll address the questions after the presentation. We have today Dr. Jorge Bernimier Iteran from the World Coffee Research, presenting about coffee breeding from a global perspective. Jorge is the breeding and technical manager for the World Coffee Research. And he, other than working for the coffee research, he has worked at INIFAP, before, in between his master's and PhD, and also did a postdoc at University of California in Davis. He got his PhD from the University of California, and he got his master's from the Hebrew University in Jerusalem. He's originally from Merida, Yucatan, Mexico, and he loves doing crosses and sailing as favorite activities. Thank you so much, Jorge, for this opportunity to hear about your research. And please, I'm going to stop sharing for you to share your screen.
Can you see my screen, Carlos?
Yes, we can. If we put it in presentation mode, that would be great. Thank you.
Awesome. Thanks, Carlos, for the invitation, and thanks to NCSU, again, for the invitation to host this seminar. Anyway, it's a little bit strange because I don't see anybody but Carlos, but I trust that there's more people hearing this. So I'm going to talk about coffee breeding for a global perspective, and so I start my talk actually on talking about the kind of like more details and nuances and just facts about growing coffee, and also from a perspective from an outsider. So most of my training and on breeding career, it has been working with annual crops, and then working with a perennial and a difficult one like coffee. It brings a different perspective to what could be done or not, So just a little bit about WCR, because I assume that not a lot of people know. So we're NGO based mainly in the US, but we have colleagues all over the world, and our mission is to kind of grow and protect and enhance supplies of quality coffee while improving the livelihood of the farmers that produce it. So in general, we kind of like help countries modernize the breeding plants and ensure varieties contribute to the profitability of farmers. Also, we get in industry feedbacks on kind of like what new varieties are needed. Also, a big part is in strengthening the nurseries, and try to keep multiple geographies of origin of coffee, so to reduce risks to the supply chain risk. So why are we here? Because caffeine is the most consumed legal and unregulated psychoactive substance, and there's a large amount of farmers, especially small farmers, that depend the livelihood on this. And most of the farms are actually in Ethiopia, Uganda, and Indonesia. And coffee is the second most traded commodity besides oil. And it has a value of export of $200 billion and the industry value of $200 billion. On the top right is just the production, the global production and consumption. And, we see somehow a dip in 2020, but probably because, I guess COVID, we're not drinking in our offices that much. And just as a reference, the largest producers of coffee are Brazil and Vietnam by far. followed by Indonesia, India, and Ethiopia. There's a variable systems of production. It's mostly in hilly areas, although there's grown in flat areas. There's which is called shade coffee, which is in agroforestry, either with or without another species as production, or only a shade. And there's also open field in hilly areas. And also there's now, there's been some like highly mechanized, mostly in Brazil, production. So today I'm mainly going to talk about Arabica coffee. So for those who are not very well versed in coffee, there's mainly two species that are commercialized, Arabica and Robusta. And so in general, Arabica is kind of like adapted to low temperature, high altitude, and kind of like mid rainfall. And Arabica is like lower altitude, high temperature, more rainfall. And Robusta is less resistance to stresses, and it has more issues with diseases. Arabica is mostly self-pollinated, and Robusta is cross-pollinated. And the caffeine content is much lower in Arabica. So again, just a little bit to reiterate the global production. And the top left one you see is the suitability for both species, and the bottom one is where it actually is grown. So you see some parts where it's mostly Arabica, and some that are mixed Arabica and Robusta. And you see, Brazil, Central America in general are large producers. West Africa is mostly Robusta. Kind of like Central East Africa is Arabica, as well as Asia is more kind of like half and half, but it's more or less Arabica and Robusta. And on the graph on the right is just to see what's the yield across during the across time and throughout the world. So you see, the global yield has been a little bit up in general, but it's mostly stagnant. And if you take the yield increase in Vietnam and Brazil, those are the ones that are actually making more progress in terms of yield than the rest of the world. So the point here is that the rest of the world has a lot to catch up to Brazil and Vietnam. And also, the effects of climate change are expected to decrease the suitability of coffee by 50% by 2050, which is, well, depending on the model. But so on the left, it's just the maps of if it's redder, is where would it be a negative effect of climate change, which is mainly on the lowland areas. And on the right, you can see the distribution. The dotted area is Arabica, Robusta, and this straight line is Arabica. And the color ones are actually the effect of climate change. So you see that, for example, in altitude, for example, the Arabica will lose quite a bit of suitability on lower altitude, and then more or less gain a little bit in higher altitude. And also we can see here that even in countries that Brazil that has high suitability currently, it will have low suitability through the effects of climate change. And for breeding, that means that we should be thinking of we should not wait until until we get there. So we have to start doing selections under conditions that will be the future conditions in the current places. Just about phylogeny. So there's about 100 species of coffee. this just to show what's the variation on flower and seeds. And what's important here is that this AFR subclade, that's the subclade that has high caffeine in general. The other species are not. And within this one, there's what I want to point out is that there's two species that has an arrow as actually the parents of Arabica. So Arabica is an allotetraploid, and thought to be the cross of Eugenioides and Canephora. And also to point out that this clade here is more or less adapted to low altitude in West and Central Africa, and here it's high altitude again in Central Africa. So so you could say that the parents have somehow adaptation to high and low conditions, altitude conditions. And just the origin, it's thought to be a single hybridization about 10 to 20,000 years ago. So it started with a single plant, you could say, and then it followed by around 200 generations of exponential growth, and then a bottleneck about 1,000 years ago. And so it's thought that most of the variation present today arose after ploidization. And then it's undergoing an expansion after a recent severe bottleneck. So you would think that the amount of coffee grown today is several-fold the number of plants higher than what it was during domestication. So an important thing is that the distribution of private alleles, so that's mutations that are just found in one individual, in one variety, in one individual, is more or less 74.4 of the variant size. And the allele frequency below 5% is in about 87%. So what does this mean is there's two things. If you want to do GWAS to map something, you will probably not pick up these variants because it's in super low frequency. Even if it has a high effect, you will not be able to just map it. And the other thing that is important is that there's also quite a bit of homolog loss during this. Since the hybridization and homolog loss, that means that you have the two genomes, the C genome and the E genome, and then one of the genomes has just a loss of the gene, or also there's silencing. And for me, coming from a background of a diploid species, it's been not difficult, but it's been it's taken time to realize how polyploids work, even if allopolyploid is somehow kind of like a diploid in some sense. So just the domestication. So on the top right here, just to point out where the Robusta and Eugenioides overlap, and so which is this circle, blue circle here, and the green circle here, and where the wild Arabica is located, which is kind of like between Ethiopia, South Sudan, Kenya, and Uganda. So this is basically it's thought to be the center of origin of the species, and it was kind of more or less like that, and so there's different theories of domestication, but the most frequent one is that in Ethiopia it was mainly harvested as a wild, as just gathering coffee from the forest, which is this old white picture here. So there was not a lot of selection. And then about the middle edges, it was it was taken to Yemen when kind of like more domestication and use arose, and then it kind of stayed like that for some years, for a couple of 100 years, which, you know, who knows? I don't know why people were not. Well, I guess they didn't want to share their coffee. Maybe that is why, you know, it's such a thing. But when there was more commerce, it was then taken to the Pacific Islands, and there was two routes. One is this route that went, some plants were moved to Amsterdam, and then I think it was a single plant that came from Amsterdam to South America. And this is what the Typica, so that's the name of the variety or the type. The Typica variety came to South America. And then here there was this other route that came to Brazil, and that's what is called the Bourbon types. And this is in the picture on the bottom right is kind of coffee coffee trees in Yemen. So just to talk about a little bit about diversity in Arabica, and if you see the graph on the left, it's just a PC of GBS data that include this Typica Bourbon type, East Africa varieties, land races, and Indian old varieties, and some samples of Yemen. And in gray, it's the survey in Ethiopia. So you could see that there's quite more, you know, still a lot of variation in Ethiopia, especially this section here that this variation is not present on the current day land races and varieties. And so this represents you know, high available resource for further breeding. And then on the right, just recently, there was a paper where they collected more samples in Yemen, which they call New Yemen. And so they ran, I think it's 9 SSRs, and found that there's this group of New Yemen varieties that are just for comparison, the stuff in the blue color is just the varieties. The red is the New Yemen, and the black is the Ethiopian germplasm. So there's all this group of Yemen that is being recently collected that could be also a valuable resource for breeding. And then in this case, this Typica Bourbon, that is quite a limited variation there here. So there was development of a strategy for genetic resources. Recently, that was published in 2017, in which WCR was part of this with the Crop Trust. And so just to show that there was different collections of coffee for germplasm, the early ones were made by FAO in kind of like the mid 60s. And this is important because there was quite a bit that actually went to the US and then ended up in CATI in Costa Rica, where there's the kind of like the largest, CATI has the largest bank, tree bank in the world. And so just as a strategy, there's now efforts to preserve and enhance these tree banks in the Madagascar and Ethiopia, Ivory Coast, and CATI, with other breeding collections. Just because probably it's something later I was going to ask, well, is the reference genome? So there's at least two of the best reference genome. One is an effort led by UC Davis, and then and so that's what's mostly it's available in Phytosome. And there's another one in which WCR was also part with a number of Italian colleagues in that's also available in our website. Yeah, you can see here the differences, but it's more or less similar features, except the technology of sequencing. So just to talk a little bit about breeding goals, and this is just for the. I'm going to try to compare at least what was different. What was different for me coming from different background. So just on the right is just going to show how crosses are made, and which is kind of like my favorite. This is, as you know, is my favorite part of being a breeder. But in general, you have to with coffee plants, you have to irrigate them to promote flowering. So you have to irrigate some and not irrigate some, and then take the pollen from the ones that you flower, and then the ones that flower, and then irrigate the other ones, and then you make crosses. So it's yeah. So for the breeding goals, yield stability, growth habit, seed yield and volume, coffee quality, biotic stresses, and abiotic stresses. So I'm just going to go through a couple of them. So for example, yield stability. So yield stability in coffee, there's two things. One is, like any other crop, you want it to be, as stable but as high as possible. So on the left is a multi-location trials that we were coordinating in different environments. And this PC, sorry for the Spanglish, but this is PC1, the environmental PC, in which here are the lower yielding sites, and here are the higher yielding sites. So what do you ideally want is something that is high and stable. Stable will be like it's, kind of like if there will not be G by E, all these lines will be parallel. But and the issue is that, for example, you have this one that have a high slope. That means that that they're kind of like responsive to high productivity locations. But what is the problem with here is that in low productivity locations, they yield lower than the other line. So ideally, you want something that is high on low productivity locations and higher in high productivity locations. So that's that's just regular yield stability. But the other feature in coffee is bienniality. That is the branches that develop now 1 year, you will be harvesting those 2 years from now. So there's and that plus environment, it gives you one high year of yield and one low year of yield. So that means that if you want to assess the yield of the plant, you need either two high yielding years, or also take into account high and low. And this is just a reflectance data, just to show kind of like how kind of like a reflectance moves from year to year. So you want, ideally you want to be, or at least most farmers want to be as stable as possible between high, they don't want a super high yielding year and a super low yielding year. Growth habit, there's two growth habits in general in coffee. There's I don't know if you can see my cursor, I hope, but you have these tall trees and dwarf trees, and this is mainly a single mutation, and is in which dwarf is dominant. In general, open field production is on mostly on dwarf now, and these high tall plants are for either mechanized. In Brazil, they use these high plants, or in forestry. And there's a lot of variation, not only height, but also branching, like branching number, position. There's a lot of variation that that goes into selection. So seed weight and yield. So a big part is also the conversion of cherries to green seed. And so this is important because it's difficult because it kind of depends on the country. Like some farmers get paid by cherries. Some farmers get paid by seed weight, and some farmers get paid by seed volume. So and that also changes between the farmer, the trader, and then the roaster. So it kind of depends on that. And so there's an issue for selection. For example, if you only do measure yield by cherry, you could be selecting for genotypes that produce a lot of cherries, but very little seeds, because probably the seeds are more photosynthetic costly. And then, so you also want to evaluate seed weight and seed volume to make sure that, you know, for example, there's countries that sell by volume. So you know, ideally you want like low density seeds, so it's like more bulky, you know, to some level. Assessing quality is also a big issue in coffee, especially because if you think what goes, you know, coming from, let's say, beans, you know, there's not a lot of pressure on taste, and there's not a lot of issues of processing. But if you think coffee, you know, you have, just imagine how much variation could be just by environment, agronomy, but then you know, harvesting, processing, storage, roasting, and then preparation. So that's a lot of points of noise. And so so the way this is done is kind of like, you know, centrally roasted, and then this cup is called cupping. So you got this professional or semi-professional cuppers. And for example, this is a cupping experiment that we did, in which there's different varieties. I just want to show like this the top one is just the variety that was overall the highest. This variety, this is the different cuppers. So just to show kind of like copper by variety interaction. And this is the one that was the lowest. The dotted green, it was the variety that had the most variation. And this red one was the one that had the least variation. So there's a lot of like copper just by sample or variety variation. That means that to get to a like kind of like a real value, you need to you know, you need to have many coppers. And then also you know, there's a lot of different features that they evaluate. Rust is a big issue, especially it was mostly present before the 1950s and 60s in Africa and Asia, but not in the Americas. While then, you know, then it was introduced to the Americas in the 60s and 70s, and having devastated at some point. So a lot of the breeding has been to develop rust resistance varieties. And just in general, the breeding areas, you know, the first layer was just massal selection in Africa and Asia. And then it went through pedigree selection from selections of these types. And then in the 70s up to now, there was integrations to Timor hybrids. And these Timor hybrids are kind of like chance hybridizations of Arabica crossed to Robusta. And so this has been used mainly as a source of rust resistance. And then lately, I mean, in the 1990s, it's been more focused on hybrid crosses with wild land races. So kind of like, you know, kind of like elite inbreds by wildland races. And the issues there's different issues with hybrids, but most farmers actually plant their new farms or the recover their farms with seeds. So there's some issues with the hybrid crosses. Just as a new sources of variation, there's several efforts now crossing Arabica by Robusta, Arabica by Liberica, and then this new synthetic Arabica, which is kind of recreating that, and then mutagenesis by EMS or irradiation. So now this is more, this was the background. Sorry for being a little bit long, but just about now we're going to talk the issues about coffee breeding today, and then what is our, as WCR, what is our response or to ideas of how to go about it. So currently there's limited cooperation between national programs and also sharing of germplasm. And this is both willingness, because at some point everybody thinks their own pure gold, and and that's not true. And then everybody needs other variation to make gains. And also it's difficult to share germplasm because it's not easy to send back and forth either tissue or seeds. There's also little to no population improvement, and the ones that have some populations is really small segregating populations. Also, there's most now have a large focus on hybrids because it's more immediate, but there's very little investment in developing new inbreds for releasing inbreds itself or for using those as hybrid parents. There's low use of markers, and also just regular long generation time, limited resources, and also limited emphasis on breeding for kind of like for demand breeding targets. So our response is how going back to the breeders equation and how to improve the breeding programs with that. So this is just a breeding by design scheme of excellence in breeding platform, and go from market studies to designing the breeding scheme, population improvement, development, and centrally is the breeders equations. And so how can we increase gain, which is having larger populations, and with that including market assisted selection and genomic selection. To increase the genetic variance is increase the diversity of populations. The accuracy, you can use high throughput phenotyping and sometimes genomic selection, and cycle length, doing genomic selection to cut short the cycle probably by half, and also speed breeding, which is with the aid of lights, external lights, or hormone tricks. And so response is a global coffee breeding network. So so this is an idea that we're building on. It's not we're actually developing this, and this is kind of like a test with a broader audience. And we're looking at developing the plans and with the community and looking for funding. So it's based on a global competition model, which includes cooperation between national programs and the normal competition that the countries have against them. But to share resources, resources as possible. And so it's based on centralized population and germplasm management, which is we call the breeding factory. And so we will share service and support, phenotyping information across global and regional and country level. I'm going to explain a little bit more about this, just the details later. And in which national programs could drive their own variety releases. And WCR would develop some lines that will be released by countries that don't have a breeding program right now. This will be based on demand, on market decisions, and also build capacity across the global building community. So part of that, which is my personal favorite part, is to actually locate and identify useful genetics. So with the collaboration with the USDA, with our colleague Dapen, we've been studying intravariety variation here is just varieties in Latin America, and we've been also doing just overall genetic diversity across all the global diversity pool, and so trying to locate clusters of diversity that will be important to include in a single breeding program. And besides this, we have this, I think I mentioned that we have multi-location variety trials. So in these locations, we're identifying which are the best varieties in terms of yield or disease resistance, or have some features that will be interesting. And we're planning to do this crossing design that I explain a little bit later, but it's basically three-way and four-way crosses between these elite lines. So you can think that national programs will, between all, we will pick the most interesting breeding lines, and we will cross all of them, in which we will give back to them after crossing, and each country will have a mosaic of everybody else's, a unique mosaic of everybody else's variation. So another part that we are actually building now with a partnership with the WCR and is building a mid-density SNP panel with mainly or USDA partner based on a DART technology to about 2,000 to 4,000 markers. This is developed mainly for genomic selection and maybe some diversity, but mainly for genomic selection. And we're kind of like piggybacking on the Excellence in Breeding platform to include this. So they're doing this in other crops of the CG system, and we're including coffee on this. And also what's important is that this improvement network should be evaluated in as much environmental variation as possible. So WCR sponsors or tries to coordinate two breeding hubs. One is the Central America breeding hub, which is the physical breeding hub is in Honduras, and the East Africa breeding hub in which the physical center is in Rwanda. But we also work with other countries that are not the ones that are darker is the focus countries, but we have, for example, in Asia, we work with India and mostly Indonesia. And so we're trying to, from all the possible sites that we could use, do the phenotyping. The one below is a cluster analysis on just the environment variable. So try to look at kind of like macro environments or different macro environments, and to try to locate in places where it will be we could get more variation on the environmental on the environment. And so so let me just this is so this slide is just to actually explain how will this work on detail. So so we will create a large population of three-way and four-way crosses, and we will, for now, we're asking funding for 7 environments for evaluation, but we might get some, as this gets rolling, we might get some different partners that will bring their own funding. Or so let's say if you have a big population of let's say 2,000 to 3,000 individuals, we will distribute to every environment. We will distribute a subset of that population to 200 to 300 plants. Will be distributed in 7 environments, in 7 different populations. So we will, and these environments will be in the different programs, national programs. So in these environments, we will genotype each plant, and then there will be phenotype. And with this, we will develop genomic selection or genomic prediction models, and there will be a larger, in what we call a factory, which will be centralized. There will be a large part of this population, and so based on the training of the selection model, we will select individuals here with the highest breeding value. And so we're doing this for several reasons, but part is that it's difficult as I said before, it's difficult to send seeds or plant materials. So let's say if you find a good plant in population 2 and a good plant in population 4, until we bring them to some site and make crosses and like that. So that will be, a couple of years. So the idea will be that just with training the genomic selection model, we can predict the breeding value on some plants in a greenhouse that could be, in Iceland, in a greenhouse, but it will not be there. But and then we can cross the selected individuals with the breeding value, then intercross and start a new cycle of population improvement. We will also bring at some point new germplasm, pass through some pre-breeding with the selected background to not go, to not bring too far of selection, too far variation that just breaks, all the good stuff that has been selected, and intercross too. And then the second part is inbred cultivar development. And so from here, we will do some kind of like rapid cycle inbreeding cultivar development. And this is what I mentioned before, tailored to the countries that don't have their own breeding program. So in this case, they could release these lines. And also each country itself will have really valuable variable population themselves. So they could also use this prediction to not only, so to add it to their phenotypic value. So they can predict based on genomic breeding value, but also phenotypic value. And they could use these plants on their own to do their own local selection. So all the different countries have the freedom to use these materials themselves, and add data to a global to the global efforts. And that's it for the talk. I think I'm 2 minutes early.
Thank you, Jorge. Thank you so much for the presentation. We are open up for questions. Again, a reminder that we'll take questions through the Q&A. We already have 1 or 2 questions here. One of the questions is, where can people find information about multi-location results if they're published?
So those trials, we have, we just condensed the data and analyzed some of that, but there's just 1 year of data on most countries on yield. Some countries have 2 years of data. So they can write me if they want more information, but we're still not publishing those because it's still not conclusive. So we don't want to like give you know, false representation of the data. But so it's not public yet, but each country has their own data, and we have that. But they can write me for more information.
Okay. Okay. And same person asked if there's an international standard for calculating yield.
No. So that is a problem. That is an issue. You know, having similar protocols for yield yield evaluation, and so that's part of what our work is. And in the case of the multi-location trials, we have a protocol. So each country does a protocol. So, but the question is no, there's variation on that.
Mm-hmm. Okay. What is the generation time for breeding? If you want to develop pure lines, for example?
Yeah. So, so it depends on, well, it depends on your breeding strategy, but it's about, so from seed to seed, the earliest that you can do, well, it depends on where and how do you do it, but it's about 3 years. So you seed, you know, you plant, and then in the year 3, you get your first yield data. And then you need 3 or 4 years of yield data to make, you know, to make your selection. I mean, if you want to select each year, so you're talking about, you know, 25 years of, you know, they're doing it the old way. But you know, I think with speed breeding and markers, we can cut it back quite substantially.
So the question here is how much variation is there in new varieties? For example, in Arabica, if you're saying that basically the release is a pure line or close to a pure line?
Well, so that, and okay, so the problem, not the problem, but the issue is that because so because of the long generation, a lot of times countries receive, and this is mainly, so this, I didn't mention this, but a lot of the new varieties actually come from cooperation with Brazil, in which Brazil sends them, you know, F4s and something like this, and sometimes they are released as F4s, F5s, or kind of bulks of F6, of F7. So there's quite so there's quite a variation on how high on inbreeding or how inbred they are, and then and also I have the suspicions that, you know, because a lot of these varieties come from these interspecific crosses, there's some kind of like residual heterozygosity that is difficult to get rid of, because there might be some I don't know if little, but some, you know, deleterious stuff that keeps passing as a heterozygote to the next generation.
Okay, here you have greetings from Paul Getz. He says it's nice to see you again. And he's asking about IPR issues between countries, particularly for the center of origin country like Ethiopia.
Hey, Paul. Thanks for attending. So Paul is my PhD advisor and mentor.
Not to put any pressure on you.
No. That's what I tried to put, you know, quite a bit. Well, not as much as possible of crop evolution and domestication. So, but that is a good point. And an issue that we have is, well, that everybody has, is that there's a lot of what was collected in the past that is in CATI or in other seed banks. It's under the plant treaty, and to use for breeding, I mean, I don't have anything against it actually for it, but it's an issue because in some corporations and some countries that you don't want to be tied to anything because it might be an issue later. But in the specific case of Ethiopia, they are not into cooperating because you know, they are the center of origin and they have that, which, you know, I understand it, you know, as coming from Mexico, I you know, for crops like maize, you know, I kind of appreciate all the effort and value that all these Mexican land races propose, but at some point, you cannot just live, you cannot just progress with land races. You need to you need to share germplasm with other people. Back and forth.
Okay. So a question here is about what are the main attributes of these new Yemen materials that you mentioned?
That is a good question. I mean, how can I say it without getting into trouble is that I think they don't know, they claim that it's, higher quality, but in general, Yemen coffee is quite high quality. And there's also this association that, it comes from a dry area, it's drought resistance. But, that needs to be proven, I think. So, yeah.
Okay. So you mentioned about generations that are needed to fix a line. It's what, 6-8 generations or what?
Yeah. So usually they're released at like F6.
F6. Okay.
Yeah.
Okay. Okay. So again, I think a question was already referred to from Ashrat. What are the main challenges for germplasm exchange? You just talked about Ethiopia. He says, are you only breathing on yield or also taste, and who decides on the parameters? And
yeah.
There's a side question on that I received through my phone is about how you measure aroma, for example.
Yeah. Well, so just one clarification. In general, WCR is we're not a breeding program in general. So we work with other breeding programs to do work. But so the question was about, so it's not only yield. So, but yield and disease resistance and all these things. But what we're trying, there's been a lot of work on quality. So part of that is aroma, and then part of that is taste and the combination. So there's been some work with Illy Cafe. They've associated some aroma compounds with overall quality. And also there's, just the cupping, kind of like the cupping phenotyping thing. And we are actually working with SCA, the Specialty Coffee Association, with Marion Peters, so to try to find a way to do this quality evaluation as high throughput as possible. But this is a goal for programs. But I think both in Robusta and in Arabica.
Okay. How many years of data you usually consider appropriate to release an inbred variety, for example?
Yeah. So ideally is 4 years. So it's like biannual. But there's some papers that say the correlation between the first high yield year is actually highly correlated with the next 3 years or whatever. So but also that could be a factor if you have you have good rains or whatever that year or not. So yeah.
And do you have input from farmers in your selection process, like participation from farmers?
Yeah. Yes, but well, WCR has we have a team of agronomists that do a lot of the the trial evaluations that we gather some information, and also through the national breeding programs that are closely close to each country's farmers.
Okay. Are there global databases? It says if there is a database record worldwide for coffee farms.
That one I don't know. I mean, there's reports on there's this. I think it's 2019. This Enveritas report on the question is how many farmers there are in the world, and that's part. That's I mentioned before. It was about I think it's like 15 million farms. So, but I'm not sure. To be honest, I'm not sure how that was collected. I don't know if it's aggregating country level data, probably.
So, question here is about the if you use pedigree breeding for your advancement of generations, and if there is a possibility of double haploid inbred development.
Yeah. So so it's what has been done mostly now is pedigree breeding, and double haploids in actually in Robusta that's possible. Because, well, it's a diploid and it's a more easier plant, but in so far in Arabica it has not been possible to do double haploids.
Okay. Here's a multiple question. That part of double haploids was part of this next question, so I consider that answer. What is the duration, typical duration of productivity when you plant a tree and when it's basically done and you had to eliminate it? And the second question of that is, are there any programs on gene editing that are taking advantage of gene editing?
So the answer to the first question is kind of depends on the variety and the location. So it goes from 12 years. So you will have to renew your plantation, after 12 years up to like 25, 30 years. So it kind of depends. Well, the variety, the environment, and the income, like the income or technology level of the farmers. And then the second about gene editing, the one that I was recently aware is there's efforts in breeding low to no caffeine Arabica coffee through CRISPR.
Okay. Ashra is asking how you address the area or location specific quality in coffee breeding.
How address what do you mean by that, sorry?
Well, probably with the effect of location on quality.
Yeah. So actually, what's happening now is we're with these multi-location trials. We're taking 7 varieties that are grown in seven different environments, where the goal for this is try to quantify, G by E, and look at how important is this. So that is a good question. And so if you ask the industry, they the lowest level that they qualify is by region. So I say, Central America, or even within region is like, Central America is, I would just say something like it's sweet and something else. And they say, Ethiopia is has different attributes. So it's not, but that doesn't take into account even variety or system. So that's anyway, it's to be determined, I would say.
Okay. I want to entertain just one more question because we only have 2 minutes and I want to respect the time of Jorge and the attendance. Ron is asking about this. It seems to be that environmental effects are biannuality or whatever you call it, also summoned to that. How you incorporate that into the model, the prediction models for genomic selection?
Yeah, so that's so there's a couple of publications from Brazilian groups that they've been there. I mean, Brazil is on the forefront of breeding, I say, in general. So there's many groups in Brazil, so they've been testing the predictions, using 1 year. So they have many years of data, but using 1 year, using 2 years, and try to or both years, and then try to predict the rest. So in our case, that's well, in general, genomic selection in coffee is really behind all the major crops. So this is like a fresh area to be research to do some research.
Well, thank you so much, Jorge. We will share the recording of this presentation, and Jorge will take also a look at the questions, and hopefully he can get back to some of the people that asked the questions that have not been answered. Thank you so much, Jorge, and thanks everybody for the attendance. Please share with me some photos or music for the beginning. If you didn't sign up earlier before the seminar started, we do play a gallery of photos and music. And thank you to Brandon, Janine, and Anthony for their help. We'll get together next Thursday at the same time. We'll have Ashra Amele from IITA talking about yam, but the yam, the Dioscorea species breeding for Africa. And it will be, I suspect, a very good seminar. I know Ashra from working in the Africa Yam Project. So look forward to that. Thank you so much, everybody. You guys have a good day and a good weekend. Okay. Thank you, Jorge, again.
No, thanks, Carlos. Thanks, everybody.
Bye-bye.